Hello students, welcome to this part 3 of clinical biochemistry question series. So let's get started. I hope you are liking this series. In this part, mainly we are gonna take cases of molecular biology, right? So what are all the cases we are discussing here? First will be alternative splicing or differential splicing. Then there's a question on sense and anti-sense strand or template and non-template strand. Then dideoxynucleotide use in PCR. Then topoisomerase question. Then SCID, that is severe combined immunodeficiency. Then a case of xeroderma pigmentosa, which is because of defect in DNA repair. Then 5' cap of mRNA question, that is very important. Then snRNA, that is small nuclear RNA in splicing. And ribosomal translocation question. Then your last question will be on mischarging of tRNA. So first question, do read the questions very carefully, right? Okay. So a scientist has inserted a particular gene in mouse model and thereafter analyzed the tissue expression. The tissue expression of that gene. Expression of that gene means conversion of that gene to RNA, right? So this is checked by northern blot analysis, and northern blot also detects RNA, okay? Then she observed 3 positive bands in lung sample, but only 1 band in all the other tissues examined. A possible explanation for this finding is which one of the following? So the gene is inserted in all the body cells, right? Genome of all the cells will be same, right? But when we have detected the RNA, then in lungs we can find 3 RNA from a single gene, from that single gene, okay? But other tissues are expressing only 1 RNA, okay? So what is the reason? This is a case of alternative splicing happening in lung tissue, okay? Alternative splicing, right? Now, alternative splicing, also called differential splicing, what happens here, that from a single gene, different mRNAs can be obtained due to alternative splicing by use of different splice site. Different exons are got together and different proteins we obtain. So let's see the detail. For example, there is a DNA segment with 4 exons. There's a DNA segment with 4 exons. This is exon 1, exon 2, exon 3 and exon 4, right? So transcription will occur and we get primary transcript mRNA where we have exon as well as intron. So this is exon 1, exon 2, exon 3 and exon 4, right? Now after transcription, post-transcriptional modifications occur. We know that, right? The basic ones you know, cap, tail and splicing, okay? That I'm not showing on this diagram. So one of the post-transcriptional modification is alternate splicing. Alternate splicing will occur so that sometimes in some mRNA exon 1, exon 2 and exon 3 are joined. In another mRNA exon 2, 3 and 4 are joined, right? In another mRNA exon 1, exon 3, exon 4 are joined, okay? So this will give rise to protein, different protein A. This will give rise to a different protein, protein B. This will give rise to a different protein, protein C, okay? So this way alternative splicing makes this happen, that a single gene will give rise to different proteins, right? Now in this question, when they say that lungs are showing 3 bands of RNA, means in lungs what is happening? Alternative splicing is happening here and we are getting 3 different mRNAs. But in other tissues, only one spliceosome is formed and only one mRNA we are getting, right? Then coming to question number 2. The figure below shows the base sequence on template and non-template strand. So this is our template strand, this is our non-template strand. What is the base sequence of RNA? This is very, very easy, but you have to be very careful also. So we know that RNA direction, the direction as well as codons of new RNA are matching with this non-template strand. This is non-template strand. Because non-template strand is same in direction with the new RNA, this is also called sense strand, means they have same sense of direction. And codons are also same, so it is also called coding strand. And one more name is given, it is called plus strand, right? Now, so it is very easy that we will take this strand, we will take direction same, 5' here, 3' here, right? 5' here and 3' here. We just have to replace T with U because T thymine is there in DNA and U uracil is there in RNA. Okay, so we will be having C, G, G, U. Now instead of this T, we are taking U. Then AAA, GC, U, A and 3', right? So in the options, direction is not mentioned. So we will take 5' on left hand side, 3' on right hand side. Okay. So your answer is A. CGG, U, AAA, GC, A, right? Then question number 3. When this kind of nucleotide is added in PCR, the image is given, what will happen, right? So this you can see what is the image given. There is a nitrogenous base, there is a sugar and there is phosphate. So base plus sugar plus phosphate, this is a nucleotide. And in PCR, we take deoxyribonucleotides. Deoxy means at 2 number position of sugar, this is 2 number position of sugar, this is 2', this is 1 number position. At 1 number position, base is attached, okay. At 2 number position, if we have OH, then it is RNA. If we have H, then it is DNA, okay. So this is correct that here we have H means it is a deoxyribonucleotide, right. Now, this is 3' position. At 3' position of a nucleotide, there should be OH. This OH is used for adding new nucleotide, But now, this nucleotide is a 2', 3' di-deoxy. Di means 2. So, at 2' also, at 3' also, there is deoxy means oxygen is removed, okay. So, this is a 2', 3' di-deoxy ribonucleotide, right? Deoxyribonucleotide, right? Now, when this is present, then next nucleotide cannot be added because this free OH group is not present. So, in PCR, at this point, DNA synthesis will stop, okay? So, from this question, you have learned that dideoxy will stop DNA synthesis and you have learned the structure of this dideoxy nucleotide, right? Then, question number 4. 2 strands of DNA have to separate for replication machinery to work on single strand of DNA. Yes, during DNA replication, unwinding is the first thing to happen. So, unwinding of DNA causes creation of supercoils. So, when unwinding will happen, then supercoils will be formed, which is relieved by action of. So, supercoils are relieved by the action of topoisomerase. So, answer is topoisomerase, right? Then let's see other options. Gyrase. Gyrase is a type 2 topoisomerase which is found in bacteria. So you can say option 2 is also correct. But out of these two, best to be marked will be option A, that is topoisomerase, right? Then option C, helicase. Helicase is the first enzyme which causes unwinding or strand separation, and this helicase creates supercoils, right? So helicase creates supercoils, topoisomerase relieve supercoils, right? Then option D, polymerase, that is the main enzyme which will do extension of the deoxyribonucleotides, that is it will add the deoxyribonucleotides in the DNA synthesis, right? Then question number 5, a 17-month-old girl suffered from recurring respiratory infections. Injection of diphtheria, pertussis, tetanus and typhoid vaccine produced only a minimal response. So when vaccination has produced minimal response, means there is some problem with the immune system, right? So less immunity, okay? Then the lysate of girls' erythrocytes was found to lack detectable adenosine deaminase activity. So adenosine deaminase, ADA enzyme deficiency. This is a case of ADA deficiency, and in ADA deficiency, which disease occurs is SCID, that is severe combined immunodeficiency. Her mother and father both showed approximately 50% of normal red cell adenosine deaminase deficiency. What is the diagnosis? Diagnosis is SCID, severe combined immunodeficiency. In these patients, there are almost no B and T lymphocytes, almost absent thymus, right? Now, what happens to B and T lymphocytes? ADA enzyme is mainly found in B and T lymphocytes, and what is the function? That it converts adenosine or deoxyadenosine to inosine and deoxyinosine respectively, okay? Now, when this reaction will not occur, so there will be increase in adenosine and increase in deoxyadenosine, okay? This is most important, increased deoxyadenosine. This increased deoxyadenosine is toxic and it leads to cell damage. Plus, this deoxyadenosine by salvage pathway will be converted to deoxy ATP. Deoxy ATP will inhibit enzyme ribonucleotide reductase. This enzyme ribonucleotide reductase converts ribonucleotides to deoxyribonucleotides. So when deoxyribonucleotides will not form, there will be problem in DNA synthesis. So cell proliferation will stop, and therefore there are absent B and T lymphocytes, right? So there is lack of DNA synthesis, especially in immune cell precursors. That's why combined immunodeficiency, both B and T arms of immunity are not working, right? Coming to question number 6, a 10-year-old girl is brought to dermatologist by her parents. She has many freckles on her face, neck, arm, hair, hands, and parents report that she is unusually sensitive to sunlight. So, sensitivity to sunlight and freckles. 2 basal cell carcinomas are identified on her face. Which of the following processes is most likely to be defective in this patient? This is a case of xeroderma pigmentosa, which is a defect in DNA repair mechanism, that is nucleotide excision repair. Nucleotide excision repair is defective here. What is the cause? Cause is UV radiation damage of DNA due to UV radiation. And what is the damage? The damage is thymine dimers or pyrimidine dimers are formed. Okay, so let's see the options. Option A, repair of double strand breaks. No. Nucleotide excision repair method is a method of single strand DNA break repair method, right? So this is not the answer. Option B, removal of mismatch bases from 3' Okazaki fragments. No, it is not a mismatch repair defect. Option C, removal of pyrimidine dimers from DNA. So yes, this is the answer. There are pyrimidine dimers are formed that are not getting removed. Okay. Then removal of uracil from DNA. No, this is not the answer, right? So question is, which of the following process is most likely to be defective? So what is the defect? That there is defective removal of pyrimidine dimers from DNA. Okay. Then question number 7. A 5' cap is added post-transcriptionally to all eukaryotic mRNAs to provide stability against degradation and create a site for mRNA recognition by translational machinery. Okay. Yes, this mRNA cap is formed and that is very useful in initiation of the translation, and it also provides stability, and it also prevents the attack by 5' exonucleases, right? Now, what is the question? Which of the following statement is not true? So you have to tell the wrong statement. Now, first is option A, 5' terminal of mRNA is capped by 7-methyl guanosine triphosphate. Yes, this is correct. There is a triphosphate bond here. So let's see this diagram. So this is the diagram. These are the 4 options in the question, right? So see the diagram first. This is 7-methyl guanosine. Methyl is present at 7th position. This methyl present at 7th position. This is guanine. This is guanine. This is sugar here. So this becomes guanosine. So this is the cap, 7-methyl guanosine cap. Okay. Now this is nucleotide 1 of mRNA. This is nucleotide 2 of mRNA. Okay. So what is the bond between them? This is 5', 5'. Usually we have 3', 5' linkages. Okay. But here be careful. This is 5', 5' triphosphate bond. Okay. There are 3 phosphates involved here. Okay. So now let's read all the options. Option A says 5' terminal of mRNA is capped by 7-methyl guanosine triphosphate. This is correct. Option B says 5' cap contains 2'-O-methyl ribonucleoside adjacent to it. So this is the ribonucleoside. This is a nitrogenous base. This is sugar. So this is a ribonucleoside. Okay. And this is having a methyl group with it. So it is 2'-O-methyl ribonucleoside adjacent to it. Okay. So this is also correct, right? Then option C, cap is attached to 5' end of mRNA through 5', 3' phosphodiester bond. No, this is wrong. And this will be our answer because question was asking wrong statement. Okay. Now have a look at option D. 7-methyl guanosine is attached to mRNA through 3 intervening phosphate groups. Yes, this is also correct. Okay. Then coming to question number 8. Splicing. Splicing is a post-transcriptional process in which non-coding sequences are removed from nascent mRNA and keep only the coding sequences exons to form a mature mRNA, right? Yes, introns are removed, exons are used in the formation of protein, right? Which of the following is involved in splicing process? In splicing process, when we have to remove the introns, which of the following is involved here? So let's see the options. tRNA, no, tRNA is used in translation, and this is a post-transcriptional modification. Splicing is post-transcriptional modification, right? So then mRNA, yes, mRNA is used, but we'll look at other options. snRNA, yes, snRNA is very specific answer. SN stands for small nuclear RNA, which is used in spliceosome formation. There is use of snRNPs, which is snRNA plus proteins. and the full form of snRNP is small nuclear ribonucleoprotein particles, right? They arrange themselves at the intron and they break exon-intron junction. Okay, so this is the answer, right? Then question number 9. A pharmaceutical company is studying a new antibiotic that inhibits bacterial protein synthesis. When this antibiotic is added to an in vitro protein synthesis system that is translating the mRNA sequence, this. So I'm writing the sequence AUG, then triple U, then triple U, then UAG. Okay, so this is the mRNA sequence for protein. So we know that AUG is the first initiation codon which codes for methionine. Then triple U codes for phenylalanine, and last is UAG, which is a stop codon. Okay, so we should be getting these 3 amino acids, right? Now the only product formed is the dipeptide formyl methionine and phenylalanine. Okay, so only methionine and phenylalanine, the first 2 amino acids are added, and then the third amino acid is not added. So what step in protein synthesis is most likely inhibited by the antibiotic? Option A says initiation. No, initiation is not affected because initiation codon AUG has worked and methionine is getting added during translation here. Okay. Then peptidyl transferase activity. Peptidyl transferase activity make peptide bond between 2 amino acids and they are joined. So we have 2 amino acids joined here, methionine and phenylalanine. Okay, so peptidyl transferase activity is also working, initiation is also working, but they are asking which step is inhibited. Now option C is ribosomal translocation. Yes, this step is affected because what are the steps in elongation of translation? That first amino acid is added at A site of ribosome, right? Then peptidyl transferase joins these 2 amino acids, right? So let's say this is P site of ribosome, this is A site of ribosome. Now after peptidyl transferase activity, we have our 2 amino acids joined at A site. Okay, now we want to shift them to P site. For that we have ribosomal translocation, which is movement of ribosome on mRNA. That must not be happening in this. That's why the third amino acid is not getting added. Okay, so answer is ribosomal translocation, right? And termination is very non-specific here, so we can just leave that. That is not the answer. In fact, in this scenario, what is happening is that ribosome is frozen before reaching the termination signal, right? And we are getting only a dipeptide. Coming to last question of this video, question number 10th. A tRNA molecule that is supposed to carry cysteine, so tRNA cysteine. So tRNA has anticodon for cysteine, so it must carry cysteine. This is mischarged so that it actually carries alanine now. So there is a tRNA which has anticodon for cysteine, but which amino acid it is carrying? It is carrying alanine. So it is carrying alanine, which is wrong amino acid carried by tRNA. It is mischarging of tRNA. Now what is the question? Assuming no correction occurs, no correction has occurred, this tRNA has gone into the elongation step of translation. So what will be the fate of this alanine residue during protein synthesis? See, during protein synthesis, there must be an mRNA where there is codon for cysteine. So this codon for cysteine and this anticodon for cysteine, they match, they can hydrogen bond. So they match and they will join. So now whatever amino acid is there on the tRNA, that will be added in the protein, and it will be a mutation. Okay. You must watch my translation video. There I have told you that the only proofreading, that is a correction during translation, can happen with the help of an enzyme amino acyl tRNA synthetase. That is when amino acid is getting attached to tRNA. Proofreading can only occur at that point. But during initiation, elongation or termination, no proofreading can occur. Okay. So let's see the options. Option A, it will be incorporated into protein in response to alanine codon. No, codon is same. Codon is same. They have not mentioned in the question that codon is changed. Okay. So this is not the answer. Option B, it will be incorporated into protein, that is alanine will be incorporated into protein in response to cysteine codon. Yes, this is correct. Option C, it will remain attached to tRNA as it can't be used for protein synthesis. No, this doesn't occur. Option D, it will be chemically converted to cysteine by cellular enzymes. No, amino acid cannot be converted like this. Okay, so answer will be B. Alanine will be incorporated into protein in response to cysteine codon. So congratulations for completing this video. I admire your dedication towards your studies. Best wishes to you.